The Ethical Side of Cloning, presented by Ursula Triplett from Embry-Riddle Aeronautical University. In 2005, the United Nations adopted its Declaration on Human Cloning, which prohibits all forms of human cloning inasmuch as they are incompatible with human dignity and the protection of human life. The issue was then given to UNESCO, who handed it to their bioethics program. It is still being debated as to how to proceed in establishing an all-encompassing global law on human cloning based on factual, unbiased evidence. 40 countries have banned reproductive cloning since 1997. China and Israel allow cloning of embryos for research only. The United States has proposed laws but has never passed any legislation regarding reproductive cloning. It was in 2013 that American scientists cloned embryonic stem cells and demonstrated that these cells could be developed into specialized adult cells that could replenish damaged tissue. Cloning until the 1990s was understood to mean plant propagation by some method other than seeds. There are three types of cloning established by the scientific community that involve human genetic material. They are molecular, therapeutic, and reproductive cloning. Molecular cloning has had little opposition because DNA is removed from and combined with other sources to create recombinant DNA. In the past 40 years, scientists have found an efficient way to develop specific, fully human recombinant antibodies. Therapeutic use of stem cell research promises medical advances that can enhance the lives of many people, but also raises many ethical questions. Reproductive human cloning has not yet been achieved. The hoped result would be a living human clone. The authors of Reproductive Cloning in Humans and Therapeutic Cloning in Primates, as the ethical debate catching up with the recent scientific advances, call for a clear-headed approach for sanctions against an implementation of reproductive cloning. Ethical issues at present are concerned with right-to-life issues. Another fact is how monies are exchanged for the harvesting of oocytes, which puts donors in harm's way. Questions of why this route would be a choice for a parent lead to questions of motivation. These motivations are argued to be in the arena of power and control over offspring. The practice of cloning also begins a change in the level of human biodiversity and opens doors to the misuse of the procedure and future organ trafficking. Therapeutic cloning requires a vast number of oocytes to be animated in various forms of research. Obtaining a prodigious amount of these oocytes requires harvesting from donors. Donors must go through a medical process that tricks their bodies into producing a mass of acceptable ova, which are then surgically extracted. After successful retrieval of the ova, these women are paid for their cooperation. This entire process jeopardizes the health of the donor and can lay the foundation for future health problems. The precautionary principle tells us that we ought not to do that which might turn out to have deleterious consequences. It took 277 attempts before Dolly the sheep was successfully cloned. This mammalian clone was known to have shortened telomeres, which resulted in an early death and was also predisposed to genetic abnormalities. Replication of cells can also be a moral ethical problem. Cells have always replicated and cells have always died. Without the attachment of human emotion, there is no argument to either type of human cloning. Legislation is more problematic than research. Funding for this type of research has been tightly limited by governments. There is a possibility that therapeutic cloning will provide a slippery slope for the overall acceptance of reproductive cloning. Therapeutic cloning provides unknown genetic outcomes, health risks to all involved in the process of extracting genetic material, and involves a huge waste of genetic material. If the huge amount of human genetic material being destroyed after project completion is taken into account, it is clearly more ethically problematic than cloning a single organism to become a live human clone. There is a possibility that some cells could be kept in a suspended animation to continually produce undifferentiated cells. This would keep the cells alive but not permit them to develop fully into their potential for human life. Such a scenario has been advanced regarding rejuvenation of aging cells. Given that an embryo has the potential to become a fully developed individual, the question arises as to how to justify the destruction of so much human potential in the name of science. When does a right to protection begin? Does the advance of scientific knowledge justify the creation of so much human embryonic cellular matter? Will the experiments conducted on this matter be beneficial to the human population? Will the information discovered justify the means in which it was obtained? In countries that have a greater religious conscience, The right to life is of deep concern. These countries have a dim view of human cloning. More secularized societies see any type of cloning as scientific advance. It took 40 years for scientists to be able to efficiently clone on a molecular level. Therapeutic cloning is still in its infancy. Human cloning, though not yet a reality, will probably be a method of reproduction in the future. I see this merely as the progress of science. Advances in therapeutic cloning should soon result in the growing of replacement organs, but at this point this would be extremely expensive, and the science is not able to generate organs on demand. This is an application of the categorical imperative because I want everyone to have healthy organs, and everyone should want that. Anyway, we have a duty to research therapeutic cloning to eventually provide healthy organs for everyone. The possibility of human reproductive cloning is a recent development in the scientific community. Molecular human cloning has taken 40 years to become efficient. Therapeutic human cloning still provide uncertain outcomes in research activities, and reproductive human cloning has not yet been achieved. Science and law seek to separate human emotion from the cloning process. Right to life questions remain. What would guarantee successful human cloning outcomes? Would human clones be entitled to the same rights as naturally born humans? Do human clones have a right to life?